Hey everybody, Mr. Regan here, and today we're going to be looking at exploring our new spider simulator to help us understand how traits become popular and common in a population, along with where did traits come from and all these new vocab words with genes and protein models and what they're all about. So our simulator today, we're going to be looking at understanding how to make changes in our spider's traits within the simulator and how to record your observations about those traits in our spider cell data table. So once we're done going through the video, make sure you come back, put your X in the row that you feel you fall under, and reach out if you need help. So in task number 3, we have two important things. We have our simulator here and we have our cell data table. And we're going to be looking to make changes to our spider to help us understand how do genes and proteins work. So when we click on the Amplify link, it's going to take us to our spider simulator. You're going to see that we have 6 unique spiders, all with 6 different names. And each spider has a lot of different traits based on different features that they all share. And you can see those features at the top of the sim. Now, each of these are different features, some that can be seen, like the bristles, that's the hairs on their legs, and some that cannot be seen, like venom. And when we click on one, you'll notice that we see these little letters here, we see the trait, and a little bit more information about the spider. And this trait not only can be seen here on this homepage, but when we click on them, we get a lot more information about this trait and what we see for this spider. So we're looking at Zora here, if you can see, and we're looking at how she has a very dense bristle, so a lot of hair on her legs. And you'll notice here on the left side, we zoom in on those bristles. And we can see those little bristles on her skin, and we can zoom in on that cell here to see what's going on inside of her body. Now, Zora's got this cell that's doing a lot of different stuff here. So let's go ahead and break down what we see. Inside of her cell, we have the nucleus, which is the control center for the cell. And inside the nucleus, we have our chromosomes. These are those purple little lines you see. Now, the chromosomes, think of them like books. And in these books, we have genes. Genes are the instructions for the different traits that we see on our spider. So, looking at our chromosome here, you'll notice that we have 2 unique genes that we're looking at, R1 and R1. So, Zora has 2 genes, R1 and R1, and we can change those genes if we wanted to. But those genes there, R1 and R1, help to make special proteins, these protein molecules. And those are the pink things you see floating around. So this R1 gene, okay, these chromosomes are our books. These are the instructions for how the cell is going to make these little purple thing and these pink things floating around. Now you'll notice here that the pink things floating around have a special shape. These look like little slides or little loops, whatever you want to call them. And there's also other proteins that they can make. And when we change our proteins, so let's say I change it to R2, you'll see the shape of those proteins change. And not only do the shape of those proteins change, if we look over here to the left side, we notice that when we zoom in on the skin, we don't see those bristles anymore. And when we look at Zora, she now no longer has hair on her legs. So if you notice, for each feature, we can select the traits. And for each of those traits, we can go inside the nucleus and go inside the chromosomes, and we can change the genes. And changing those genes affect the proteins that are important for deciding our traits. So here's what you're going to be doing. Similar to what you just saw, you're going to select an observable trait for one of the spiders, any spider that you want. You need to pick a spider that you feel is best. From there, you're going to record the name of the spider. You're going to record the genes it has before you make changes, what the trait is. So I would have put Zora here. The genes were the R1 genes, and the observable trait was lots of hair, lots of dense hair. The protein shape was that pink slide-looking shape. And after the change, I'm going to record that new information. So R2, the observable trait is no hair, and the protein shape was now that big, bulky circle. I'm going to do that for one trait, and I'm going to do that then for a second trait so that we can look and see how do genes and protein molecules affect traits. If you have questions, reach out. Otherwise, complete your survey and good luck.